Good morning, folks, or perhaps good afternoon, good evening, or good night, depending on when you're watching this. A little bit of a change of scenery today. I am in one of my son's room because it's right now the quietest room in the house. So we want to talk today about cell transport, stuff coming across the cell membrane, nutrients coming in, waste coming out. There's a number of different ways that the cell can go about doing these. Some require energy, some of them don't. So we want to spend some time talking about that, the different examples, and then we'll have some recap stuff available for you at the end if you'd like to use those. So without further ado, let's go ahead and get started. All right, so we've got cell transport, and the primary organelle that we're going to be talking about when we're doing cell transport is the plasma membrane, right? That's that phospholipid bilayer that surrounds the cell, the hydrophilic head, phosphate head, hydrophobic tails that provide a barrier. Now we have to get nutrients across this barrier into the cell. We also have to get waste outside of the cell and other products like hormones and stuff like that. Energy molecules in the form of glucose have to get out. And so there's a particular characteristic that cell membranes have. It's called permeability. And permeability is what determines if things can come in or out or not. Now, if nothing can go through, it is impermeable. So think like your skin, for example, okay? If you splash water on your skin, water can't come in, and water's not going to be able to come out through your skin. Now, there are pores where some of that stuff comes in, but that's a little bit different, and that'll get into some of the conversation that we're going to have a little bit later about different types of transport. If anything can pass through, it's freely permeable. So think of like a piece of fabric, okay, like a shirt, right? Water can come in and out freely. The shirt's not going to stop it, okay? So that's freely permeable. If movement is restricted in some way, then it's selectively permeable. So a plasma membrane is selectively permeable, which means some materials are going to come across freely, but other materials can't. Now, characteristics that restrict movement is going to be the size of the molecule, the electric charge, the shape of the molecule, and if it's soluble in lipids, those oils and fats and stuff like that. Transport through the plasma membrane can involve two processes: passive processes, there's no energy required, it happens spontaneously, it's another way to describe it, or active processes which require energy. Those typically, that's kind of like rolling a ball uphill versus a ball downhill, right? Both of them are transport. One requires energy, one of them doesn't. Some examples of the different transport processes we have are passive transport is diffusion and osmosis. Diffusion and osmosis are basically the same. It just has to do with what is actually doing the moving. Something called carrier mediated transport. That just means there is something allowing the molecules to cross the membrane. That's what the carrier is. Now, it can be active or passive. And we'll look at those in just a second. And then something called vesicular transport, and that's active. And that's using vesicles, which are a type of cell organelle, but they're typically temporary, okay? We'll look at that a little bit. That's your endo and exocytosis, basically. So different things that affect diffusion, okay? If things are going to speed it up or slow it down. How far the particles have to move, the shorter the distance, the faster it takes place. The molecular size and the ion size, the smaller the molecules, the faster the diffusion, because they're basically, think of diffusion as kind of like having to navigate through a crowded room, okay? If it's really, really crowded and you're small, you can get through very, very quickly. If you're large, like a grown-up, it's more difficult to get through. The temperature, the hotter or the more heat, the faster the diffusion because everything's moving faster. The concentration gradient, so that's how much stuff. So think of a drop of food coloring in water, right? The concentration gradient is really high of the color in the drop. And so it's going to spread very, very quickly, and then it's kind of starts to slow down as it moves farther and farther away. And then the last one is the electrical forces. So if you have like charges repel, opposites attract. So if you have opposite charges, they're going to move faster towards each other, and the opposite will happen if they're apart. So here's a diagram of a standard cell membrane. We've got our phospholipids right here as part of the plasma membrane. Now there are some things like right here, the lipid-soluble molecules will actually pass through the membrane without any help. They just sort of slide right through it like water through that shirt. Okay, so they're going to come through without any help. That's just regular passive diffusion. We've got other things that will dissolve in there are simple gases like oxygen and carbon dioxide. You don't have to have special molecules to move that stuff through. And water as well will pass freely. Here's our little water. Will pass through that cell membrane freely. Now, channel mediated, if you remember a couple of videos ago when we talked about the plasma membrane, we talked about embedded proteins, integral proteins, and surface proteins. And so some of those proteins are channel proteins. Basically what they do is they open up a channel. Here's an example right here that allows molecules to pass through. It's still passive transport, so it's still diffusion, but they have that path to go through. So this would be kind of like a door in the cell membrane that allows stuff to pass through, but only at that point. So things that go through there are water-soluble compounds and ions, like sodium channels and potassium channels, for example. Their movement through these channels is going to be affected by the size of the particles, the charge, and how they interact with the channel walls themselves. So that's just regular diffusion. Now regular diffusion, let me come back here. Regular diffusion is the passive movement of solute from high concentration to low concentration. Okay, so I've got a little bit of a demo here for you. I've got a jar full of water, okay, and then here's our food coloring, right? And so diffusion is the movement of these particles from high concentration to low concentration. So I want take a look, and right now when they first get in there, you can see that's high concentration. You can see them, they're starting to spread out, right? Let me get out of the way so you can see this a little bit better. So they're going from high concentration to low concentration. So when they first hit the water, there was very high concentration. The low concentration would be low concentration of the color in the water right here. Okay, so what's happening is we're moving down that concentration gradient until we end up with the same concentration of color all throughout this liquid right here, okay? We're going to take a look at this here in just a second so I can show you, again, what that really looks like. All right, so now if you take a look, you can see it's more of a pink, right? Here, I've got a piece of paper right here that can sort of show that off. So it's definitely more of a pink. If you look down here at the bottom, we've still got a little bit of a higher concentration of food coloring down there, and that's just a density thing. The food coloring is more dense than the water, so it sank. But we've got a more even distribution. So now diffusion is going to be slowing down throughout this. Now, if you want to speed up passive transport, right? Smaller molecules, which can't really do anything about. Temperature, if we heated this up, it would speed up faster. Also stirring. So that's why we stir, like if you're making Kool-Aid or something like that, you stir it to make sure that it gets evenly distributed. You could also heat it as well. That makes it go through faster. So here's our passive transport. And that's just regular movement of the particles. And then just remember that osmosis is movement with a membrane involved. We don't really have anything helping it out. It's still very passive. And so that's diffusion. Osmosis works the same way, but it's a special kind of diffusion. And so this is the movement of water, and there has to be a selectively permeable membrane separating two solutions with different concentrations. So if you look at this picture right here, we got to look at the dots. Okay, so the water molecules are the little blue dots, and they're kind of small, right? Water is a very small molecule. And then the solute, let's call it sugar, okay, or salt, something that you dissolved in the water to make your solution. Those are your bigger molecules, okay? So we have those, and then here's our selectively permeable membrane right here. And if you notice, this works the same way, but it's a special kind of diffusion. And so this is the movement of water, and there has to be a selectively permeable membrane separating two solutions with different concentrations. So if you look at this picture right here, we got to look at the dots. Okay, so the water molecules are the little blue dots, and they're kind of small, right? Water is a very small molecule. And then the solute, let's call it sugar, okay, or salt, something that you dissolved in the water to make your solution. Those are your bigger molecules, okay? So we have those, and then here's our selectively permeable membrane right here. And if you notice, if you look really, really closely, the arrows indicate that the water can pass through, but the solute can't. So normally what would happen, in order for the concentration to equalize, the solute moves, and that's just regular diffusion, okay? If you put that selectively permeable membrane, only certain things are going to be able to pass through it. And so what ends up moving then to balance that solute concentration is the water. And so as we have one side has a higher solute concentration, right? It's got more of the purple bubbles. That's going to mean that on side B has a higher water concentration. And when I put brackets around it, that just means concentration. Okay, think back to chemistry. So B has a higher, no, that's wrong. I had it backwards. B has a lower water concentration, okay? So that means there's less water on one side than the other, and that's what's doing the moving. Higher solute concentration, less water concentration. So what's going to happen is the water is going to move, and so you'll actually end up with a change in volume until they equal out, okay? So what we're looking at is the total number of molecules, okay? Once the total number of molecules equalizes, you'll end up with a volume change between one side and the other. And so this ends up creating some really interesting phenomena that takes place, especially with cells. And we're actually, the lab that we're going to be doing in class is going to be looking at this phenomenon. And you're going to use an egg, which for all intents and purposes, we're going to create one giant cell out of that egg. Now, it's not just one giant cell, but the analogy works. Okay. So we have this movement, and if it's allowed to happen freely, we do have volume changes. One side has a little bit less volume, one side has a little bit higher volume. And so we can also talk about something called osmotic concentration, which is the total solute concentration in a solution. And tonicity is going to describe that solute concentration. And so there's three ways that you can describe it. There's isotonic. Iso means equal. So you have the same amount of solution on both sides of the cell. This is a picture of a red blood cell. Here's an actual photograph of a red blood cell. And if you notice, they kind of have a little disc shape in the middle. They look kind of like shallow bowls or something like that. And that little cup is where the oxygen sits, okay? And actually, we'll talk about this next semester. The nucleus is ejected at the last step before the cells are released. That's why you are constantly replenishing your blood cells. They only last for a certain amount of time. They don't have the capacity to make more of themselves. And so they look normal in size. We have equal movement. If you look at the arrows, we have equal movement in and out. So what we end up is no net movement is what we say. There's no change. If you have too much water coming in, that means your cell is in a hypotonic solution. Hypo, we've kind of already talked about. Hypo is low or beneath. So what you're doing is you're putting your cell into a solution that has a very low solute concentration. And what's going to end up happening then is your water is going to come into the cell because there's more solute inside than outside. We want to balance that solute concentration. So the water is going to come in. That's going to make your cells swell up like a balloon. And you take a look at that picture right here. The little cup is gone. It's filled in at this point, right? And if it's too hypertonic, what will end up happening is your cell will break. The cell membrane will rupture, which is also known as lysing. So you can actually have your cells burst, which is really bad. And so this is one of the reasons why you shouldn't drink a lot of water really fast. There was for a while, there were radio game shows where whoever could drink the most water the fastest would win a car or something like that. But you can really cause a lot of damage if you do that. So don't drink water. You want to drink a lot of water, but drink it throughout the day. Don't do all three. Don't do like 3 gallons all in one shot. That's not good for you. So then going in the other direction, you put your cell in something that has a whole lot of solute. That becomes hypertonic. Hyper is high or above. So we've got a very high solute concentration. That means there's more water inside than outside, and we want to balance that concentration. So all the water is going to leave the cell, and that's going to make your cell shrink like this, okay? And that's going to make things very difficult. The cell isn't going to be able to do the processes because there's not enough liquid, so that's going to slow down diffusion and stuff like that. And the cell could very easily starve to death or be poisoned, basically, because there's an accumulation of waste. So isotonic, it's equal. That's what we want, okay? Hypotonic, if you put a cell in a hypotonic, your cell swells up like an hypo. If it's hypertonic, right, you're really hyper, you've got a lot of energy, and then once you burn all that energy, you're just like, and you kind of deflate, right, and you shrink. So if you're hypertonic solution, your cell is going to shrink. Now, I will say you have to keep in mind when we're talking about hyper, hypo, and iso, we're talking about the solution outside in relation to the contents of the cell inside. So the solution you put the cell in is iso, hypo, or hyper. Okay. We can also refer to, so like if you put a cell in a hypotonic solution, that means the cell is hypertonic to the solution, right? It's backwards. And same thing with hypertonic. If it's a hypertonic solution, the cell is hypotonic to the solution. But we're always comparing to the solution outside. So now what we get into, that's when we get into the facilitated transport. And those can still be active or passive. So carrier mediated transport, it's also known as facilitated transport. Basically what we have going on is we have something helping this movement process, okay? And typically it's proteins that are moving this across. They could be passive like channel proteins. It could be active processes like ion pumps or glucose pumps or something like that. Typically, they're very specific. There's specificity. So one type of protein is going to move one type of solute. You don't really have a whole lot of like one size fits all sorts of things. A sodium channel is only going to move sodium. A potassium pump is only going to move potassium, that sort of thing. The rate of transport is going to depend on the saturation limit. So saturation would be would refer to the number of proteins, and substrates would be whatever you're moving. So let's stick with the sodium ion channel. The substrate is what you're moving, so it's the sodium. And basically, if you have a lot of sodium, but not a whole lot of channel proteins, we say it's saturated. And so that's going to sort of slow things down. There's going to be a bit of a bottleneck because we basically filled up all of our capacity to move stuff. The other thing that we look at is regulation. So hormones will affect the ability of carriers, open them up, close them down, stuff like that. So different types of transport. There's 3 different types of transport. It has to do with the direction that stuff is coming across, basically. So the first one is uniport. So port has to do with like the channel, okay? Uniport, the uni is one, like a unicycle. So they only move in one direction. This is your one-way street, okay? Now, it's still always only going to be one substrate, one thing that's moving, but in this particular case, it only moves it one way. When we talk about nerve impulses, we'll look at sodium and potassium ions that only let stuff in or let stuff out. So that would be an example of uniport. Then we can look at symport. So now we're getting in 2 substances at the same time. Sym means the same. So if you notice, they're going the same direction. We've got 2 substances going the same direction at the same time. And then the other one is antiport. Antiport, also known as countertransport. Now, symport can also be called cotransport, and that's really important because a lot of times, let's say there's one specific thing we want to move, but in order to move it, we have to move something else. And so the goal is to move, like maybe the goal is to move the purple, but in order to move the purple, we have to move the blue. That's co-transport. They're going the same time. Antiport, which is also known as counter-transport, is moving substances in different directions. Now, again, this would be if we want to move purple in, but in order to do that, we have to move blue out. Okay. Now, these can be, like I said, a couple of times at least, this can be active or passive transport. We've also got facilitated diffusion, which is specific carrier proteins that will move things that are too large to fit through the regular channel proteins or something like that. And so they will, these are like gates, basically, gated channel proteins or gated proteins or something like that. And so basically what ends up happening is you have a molecule, like this is glucose. It's going to bond to a receptor site on this protein. If you notice, if you look in right here, there's a little like half hexagon pocket that glucose fits into, because glucose is it's the only thing that you put into your body that's hexagonal shaped. Other molecules have different shapes, and so they don't fit there. And so there's like half of a hexagon pocket that fits there. Once it sits into that little pocket, then the protein activates basically, and so we go to this side. Now, if you notice, it switches places. So it starts out like this, and then the protein fits right here. Here's my little glucose, right? Once the glucose is in there, my hands are going to go like this because the protein's going to open and it's going to enter into the cell, right? And so there's your facilitated diffusion. They're too big to fit through channel proteins, because channel proteins, remember, they're small charged ions or molecules. sodium and stuff is going to pass through that. If it's too big, we use our specific carrier proteins. So still with carrier mediated transport, now we have active transport. So active transport, which is what we're talking about right here. Remember, if you want to be active, right, you have to have energy. And so when we're talking about cells, ATP is the energy that we're talking about. So active transport requires energy. Now, a couple of things about active transport. Active transport moves against the concentration gradient. So normal diffusion, remember our cup with the food coloring, that goes from high to low concentration. This would be like rolling a ball down a hill, okay? What do you have to do to roll a ball down a hill? Push it. So it needs a little bit of energy to get started, but once it gets started, and so this would be like adding your drop of food coloring into the water. Once it gets started, what happens? Right? It rolls down the hill. You don't have to do anything after you put that initial drop in there. It's going to spread out on its own, okay? Active transport is different. Active transport is against the concentration gradient. So you're going from low concentration, the bottom of the hill, to high concentration, which is the top of the hill. And so now, if you've ever tried to move something up a hill, yourself, a ball, something like that, push a cart, you've got to put energy into it, right? It takes work. You get tired at the end, especially if the hill's really high. So that's active transport. It requires energy to do that. Okay, and that energy is ATP, adenosine triphosphate. It's derived from adenine. It's kind of modified a little bit, and you put all these other phosphates and stuff on there. And so we've got, typically these are going to be our pumps. Okay, so like ion pumps, this is, let me back up. So an ion pump is something that pushes ions like sodium or potassium against the concentration gradient across the cell membrane. Now, it's only going to pump these one things. There's also calcium pumps in muscles and stuff like that. It's only going to pump one thing, right? So this is uniport. It's either going to pump it in or out. You can also have exchange pumps. That's what this one is, right? And so we've got things going in different directions here and here. And so that is our antiport, our counter transport. They're moving in opposite directions. We are exchanging 2 potassium ions for 3 sodium ions. That ends up changing the charge across the membrane, which is super important with nerve impulses, and we'll talk about that like November-ish. Okay, 2 different kinds of active transport. We've got primary active transport. Okay, primary active transport, you're moving the substances directly. So like this potassium ion, sodium potassium exchange pump, you're using ATP to directly move stuff. Okay, so this is your water pump. You plug it in, your pressure washer. Okay, so pressure washer, you take your hose and you put it into basically a motor, right? And then that forces the water through more quickly. So that's primary active transport. The pump is acting directly on whatever substrate we're moving across, right? So then the other one is secondary active transport. And what that ends up doing is you have a pump to establish a concentration gradient like you do with the primary. But this is so something else will move. So take a look at the example I have on the screen right here, okay? We've got our sodium potassium pump right here, okay? And it's taking out sodiums, it's bringing in potassium. That's going to be setting up a concentration gradient. The reason why we're doing this, okay, is not just to change how much sodium and how much potassium that we have. It's to start bringing glucose in. Now, how that works, okay, so we set up a concentration gradient of sodium outside of the cell. If you take a look right here, we've got places for the sodium ions to sit. Remember, we set up a concentration gradient of the sodium ions. So now there's more sodium outside than inside. So it's going to want to come in, high to low concentration, right? Well, it's going to bring its friend glucose with it. And so while the sodium is coming in, the glucose is also going to come in. And then eventually that concentration gradient is going to resettle. So we've got the same amount of sodium on both sides. So then that sodium potassium pump is going to kick in, active transport. It's going to use energy to move that sodium back out, reestablishing that concentration gradient to bring more glucose back in. Let's see, a little bit more. Let's keep talking about active transport. Now we're doing vesicles and bulk transport. So vesicular transport, vesicles, right? Vesicles are basically just like little sacks or bags or boxes, bottles, whatever you want to call them, right? They're typically like little pouches made by the cell membrane right here, okay? The cell membrane kind of bloops in and then makes a little bubble, and then the bubble breaks off. Or it happens the other direction. You have the little bubble, it sticks to the cell membrane, it becomes part of the cell membrane, and it just goes boop, and it pops the stuff back out. That's getting into endocytosis and exocytosis. And now, as far as endocytosis is concerned, there's three different kinds, okay? So there is regular endocytosis. So that process I just described, I looked at, that is receptor-mediated endocytosis. So it's determined based off of if these receptors get triggered or not. Okay, so if there's no if the receptors don't get triggered, it doesn't happen. Okay, you've got pinocytosis, and that's endocytosis, but that's moving water, basically. So like this vesicle right here basically drinks water from outside of the cell and then brings it in. So it's bulk transport of water, basically. And then the other one that you can have is phagocytosis, and this is basically the cell eating something really, really large. So there's a special type of white blood cell called a macrophage that will actually reform itself to spread around a cell, a large something, and then basically those lysosomes and peroxisomes break down that something. And so macrophages will actually surround like a bacterial cell, because bacterial cells are way smaller than human cells. And so this macrophage is like a big giant monster. It's just going to come and it's going to eat. It's going to move around and it's going to eat basically that bacteria, and then there's lysosomes and stuff in there, and digestive enzymes are going to break that bacterial cell down and destroy it and recycle it, basically. And so that's phagocytosis. Now so those are three. Endocytosis. Endo is inside. All right. The other one is exocytosis. Exo means outside. Bugs have an exoskeleton. Their skeleton's on the outside. So this is putting stuff back out. Okay. Exocytosis, putting it out. Endocytosis, bringing it in. Okay. That basically does it for us. I've got a few slides in here. Feel free to, you can go back and look at the lecture slides to download these ones. They're handy dandy little charts for looking at the different processes. Like diffusion, which is passive transport. either within a cell or outside of it, going from high to low. Look at different factors like size, the size of the gradient, the size of the molecule, temperature, stuff like that. These are going to be gases, small inorganic substances, lipid-soluble materials, and stuff like that. Osmosis is the movement of water across a selectively permeable membrane to try to balance solute concentration, right? And only water is moving. Facilitated diffusion is using proteins to help move stuff. Active transport is using energy. Endocytosis is bringing stuff into it. You've got receptor mediated. There's a triggering mechanism. Penocytosis is water. Phagocytosis is bringing in very large substances. Exocytosis is ejecting large amounts of stuff. So, for example, waste. Large amounts of waste, they are removed by exocytosis. That basically does it. Go back through and take a look at this. If you're having any questions, make sure you get the highlights. What's the difference between active and passive? The tricky part that folks tend to have trouble with is the hypo, and isotonic. Isotonic is the same, no net movement. Hypotonic, more solute outside, so the water comes inside. You get a big O, your cell swells. Hypertonic, all the water's going to leave, so it shrivels up like a little raisin. Keep these things in mind and have these notes handy when we're taking a look at the lab, so you can keep all that stuff handy. Maybe look at the question of why it's really important that doctors have the right solute concentration for your IVs, and what the big deal about the phrase is water, water everywhere, and not a drop to drink. If you have any questions, let me know. Send me an email, send me a reminder, ask me in class. Otherwise, I look forward to seeing you all again soon.